Thank you for accepting our invitation for this interview.
Definitely.
I'm very honored to have you today. I think you are the pioneer in the field of molecular neuroimaging, applying to acupuncture and chronic pain, including fibromyalgia. Firstly, the audience are not familiar with the molecular neuroimaging. Can you tell me the brief introduction for what is molecular neuroimaging is?
Sure. There's a couple different techniques that researchers use to investigate molecular abnormalities or pathological factors in chronic pain patients, as well as treating them with different interventions like acupuncture or acupressure to see how that affects neurochemistry. The two main techniques are positron emission tomography, which uses radioactive molecules and assesses either neurotransmitter levels or receptor binding ability. So that's positron emission tomography, or PET. The other chemical imaging technique is called proton magnetic resonance spectroscopy, and it doesn't use radioactive substances, so you can do multiple sessions with the same individual without having the problem of radiation accumulation. So proton spectroscopy is really nice for that, and with proton spectroscopy you typically are looking at either glutamate or GABA, the brain's major excitatory or inhibitory neurotransmitters respectively. So those are the two main techniques.
So I'm wondering what brought you to the acupuncture research?
So my basic training is in electrophysiology. I started out as a graduate student in an electrophysiology lab, and I studied ion channel kinetics and permeation properties of ion channels. So I was doing a lot of basic science for my PhD. But during that time I started learning meditations, things like Qigong and Tai Chi and martial arts, and I was really interested in how those things worked because I felt that they were changing me both physically and mentally and emotionally, and I wanted to try to understand that. And so I got interested in acupuncture because I thought acupuncture would be a controlled way of studying it, and it turned out that there was a lab in Washington, D.C., where I was learning acupuncture. There was a lab that had a clinical trial. They had a grant to study fibromyalgia patients with acupuncture, and they needed a research assistant. And so I just joined the lab and became a research assistant and performed acupuncture research on that clinical trial during the day, and then at night I learned how to do acupuncture clinically to treat patients at the same time, and so that was how I got involved. So it was very serendipitous. I didn't really plan it out that way. It just happened, and I was very, very fortunate and lucky.
I know you have been a lot of beautiful research and published a lot of beautiful papers. And can you give us what is the most important findings you've done in acupuncture research?
So I'd have to say I haven't published a lot in acupuncture, but I've published a little. One of the studies which I think has gained a lot of attention was my mu opioid receptor PET study in fibromyalgia patients, where we found that sham acupuncture and verum acupuncture induced the same analgesic response, but the mechanisms by which they engendered that response seemed to be quite different. The sham acupuncture seemed to increase the release of endogenous opioids, whereas the verum acupuncture somehow was able to change the receptor binding ability, maybe through increasing receptor number. So that one I think has probably gotten the most press. I've also been involved in the clinical trial that I mentioned earlier that I was the research assistant of. That one has actually gotten some nice press as well over the years, and we basically showed that it didn't really seem to matter whether the needles were stimulated manually or if they were in the correct locations. There seemed to be a nice analgesic response in fibromyalgia patients. And then I've also done a study looking at proton spectroscopy, showing that when acupuncture changes glutamate levels in the brain, clinical pain and experimental pain also change as well, and there's a nice correlation, and they track well with each other.
So you perform a lot of the different mechanisms between mere acupuncture and sham acupuncture. But however, recently published many clinical trials failed to show the differences between mere and sham acupuncture. What do you think the meaning of these lines of studies are? And then, can you tell us the suggestion to overcome this kind of problem for the future research?
So, a couple of things. So, we now know that acupuncture for chronic pain does have a specific effect if the needles are put in the correct acupuncture point locations. But that effect is quite small. So if you look at acupuncture trials for knee osteoarthritis or chronic low back pain, both acupuncture and sham acupuncture reduce pain significantly. The added benefit of correct needle placement gains you only about an effect size of 0.15, which is a small effect size, but it's statistically significantly different. So I wouldn't say that there's no difference clinically. There is a difference clinically, but it's just kind of small. You have to treat a lot of patients to really see that effect. One thing I think we might be able to do better is a lot of times in chronic pain studies, the patients themselves are very heterogeneous. So we don't really understand the different pathways for development of chronic pain. So for example, in fibromyalgia, there could be a whole number of different problems in the patients that might cause them to have chronic widespread pain, but they don't all have the same pathology. And so we're learning that now, that there's actually subgroups of pain patients, even under the umbrella of like fibromyalgia or chronic low back pain or knee osteoarthritis. They're mixed. So I think one thing we could do in acupuncture studies is if we have a more homogeneous or more controlled population that goes into the study, that reduces the variability and increases our likelihood of detecting a difference between Verum and sham acupuncture. That's one potential way that we might be able to improve efficacy studies, clinical trials looking for efficacy for acupuncture. Another way may be to look at quantitative sensory testing and use quantitative sensory testing, this ability to determine how sensitive someone is to an experimental pain stimulus, and use that to predict responsiveness. We did a study that was published last year where we looked at the fibromyalgia patients and we squeezed their thumbs with a pressure device, and we found that the patients that were not very tender didn't respond to sham acupuncture very well at all, but they responded to Verum acupuncture quite well. So if you actually stratified the population to like not so tender people versus very tender people, if you just looked at the non-tender people, you would have had a significant difference between Verum and sham, because for some reason the fibromyalgia patients that are not sensitive to stimuli don't get an analgesic benefit from sham. So that's another way of, so again, it's kind of getting at that subgroup kind of hypothesis.
Thank you for your suggestion. I think it is very helpful for the acupuncture researchers.